Hi, everyone, and welcome to Mathematical and Computational Modeling of Cancer seminar. I'm very happy to have Kamila Naxerova here today. She is an associate professor of genetics at Harvard Medical School. She did her PhD at Harvard University, and she did her postdoc with Stephen Elledge at Harvard Medical School. And I'm very happy to hear her talk today about selection in cancer evolution.
Great, Ivana. Thank you so much for inviting me. It's great to see you again. I've known Ivana for many, many years since I was a small grad student and she was a postdoc. So it's a really nice opportunity to reconnect here. As you can see,
Sorry, Kamila, just before you start, I forgot to mention for the audience, please let us know if there's any technical difficulties, and feel free to ask questions by posting them in the chat, either during the talk or at the end of the talk.
Perfect. Yes, please ask lots of questions. I'm happy to take questions in between also, because I always feel like it's better to resolve any issues right on the spot. And as you'll see, many parts of my talk are logically connected. So, if you lose the logical thread early on, definitely worth, asking questions. So the title of my talk today is Disentangling Correlation and Causation in Somatic Evolution. And that's a lot of big words. What do they all mean? Hopefully after a bit of an introduction, it'll start to make sense. And so, for the people who may be coming from very different fields of mathematical modeling of cancer, I'll just introduce very broadly, what is somatic evolution? It's really just Darwinian evolution. So mutation, selection, drift, and migration, but as it happens among the cells in our bodies. And my lab in particular is really interested in the entire trajectory of somatic evolution. So starting out with how mutations arise in the cells that sit in our normal tissues, how these mutations, if they confer a selective advantage, may expand over time, what determines the rate at which they do this is a particular point of interest that I'll be touching on today quite a bit. And then as we come to understand more and more now, cancer really arises on the patchwork of these mutant clones that are populating our aging tissues. And, it's a really interesting question, what genetically divides a cell that eventually becomes a malignant lineage and a cancer from just a mutated normal cell in our tissues. And that's something that we're really interested in. And then also, of course, somatic evolution doesn't stop once the malignant lineage emerges. There's the big and interesting field of primary tumor evolution, looking at the dynamics of that. And then finally, one area in which my lab is invested a lot, but I won't be covering today, is metastasis evolution. So how do metastatic lineages emerge from that evolutionary process that is happening in the primary tumor? So that's really the entirety of our lab's interest. But today, I'll be really talking more about the left part of this diagram. So in particular about the process by which mutant clones expand in our normal tissues, and then the crucial step from a mutated normal cell to a cancer cell. Can you see my cursor? Is that working? If I move it?
Yes.
Okay, perfect. Okay. So when it comes to somatic evolution, it's really a new and emerging field that has really benefited from huge advances in sequencing technology that have been made in the past 20 years or so. But in many ways, like a lot of the thinking that's behind it, of course, takes its clues from germline genetics. And so when it comes to the question of correlation and causality, which is what I really pointed at in my title, I just want to talk a little bit about the differences between the way we think about causality in germline genetics versus in somatic genetics, because it's a really, really interesting area that I think is maybe not getting enough attention so far. So causality is really clear in germline genetics. If you do a proper statistical analysis and you find there's a group of patients that have a particular phenotype, for example, a disease, and you sequence them and you find that they consistently, after adjusting for all possible confounders, have a particular mutation, while the control background population, people with no such phenotype, don't have this mutation. It's pretty clear that a reasonable hypothesis is that this mutation, this germline mutation, causes the disease, because reverse causality, the disease causing the germline mutation, is impossible. The germline mutation is pre-existing, so it temporally and in many other ways, it's very clear that the germline mutation must be causing the disease and not the other way around. However, in somatic genetics, causality is really much more difficult to understand. Of course, there's always the possibility that the same type of connection exists, namely that you have a somatic mutation. So that means a mutation that's not present in the germline, but only a fraction of our somatic cells. So a mutation that arose at some point after the zygote existed in embryonic development or later in life. And it's absolutely possible that such a somatic mutation can cause a phenotype, including a disease. However, if we see associations between somatic mutations that we measure in humans or in other settings, but mostly in humans, in somatic mutations, we always also have to consider there's other possibilities of causal arrows between these conditions. So because diseases and really many other lifestyle factors and exposures really change the way evolution happens in our tissues, it's also entirely possible that the disease causes the frequency of a somatic mutation to increase, or even causes the somatic mutation to exist. So we have to consider this causal arrow. And then, of course, there's also the possibility that there is a hidden causal factor that independently causes the disease and the somatic mutation. So as you can see, in somatic genetics, causality is a little bit more complicated. And I think our field, so specifically genetics, I should probably explain that I'm a geneticist. I'm not a mathematician. I love to work with mathematicians. I'm very inspired by all of you guys. I'm not a mathematician myself. So in my field, in genetics, I feel that the complexity of the possible causal connections between somatic and genetics and disease in particular is really still an emerging field that would really benefit from rigorous quantitative thinkers like mathematicians and presumably parts of this audience. And so I hope that if there are maybe people listening who are interested in this area, I strongly encourage you. I feel that we need you. The somatic genetics field needs you. So what I want to do today is basically, after this broad introduction, show you two examples. There's two different studies that kind of shed a particular and probably interesting light on the causal connections between disease and somatic mutations that are a little bit counterintuitive. So essentially, what you'll be hearing today are two examples of this very complex connection of causalities in somatic genetics. Okay, great. So the first part that I'll be talking about is what explains the association between clonal hematopoiesis and cardiovascular disease. And I just want to start out by acknowledging the people who did this work. So this was a really cool collaborative project that was led by Alex Heide, who was at the time a Harvard graduate student, and then two postdocs from MGH, David Rode and Cameron Alpine. And so you'll see this was a really nice collaborative effort where you have quantitatively trained people, mathematically trained people working with biologists and clinicians to try and address a question. So just in case you have not heard of clonal hematopoiesis, clonal hematopoiesis is a condition that a lot of people are interested in these days. And it's really just a condition whereby a good fraction, a measurable fraction of your peripheral blood descends from one particular hematopoietic stem cell. So typically, our blood is a collection, a polyclonal collection of lineages that descend from about 100,000 hematopoietic stem cells that we all have in our bone marrow. And really, each one of these 100,000 hematopoietic stem cells should be contributing a small fraction to the blood, maybe not all at the same time, but they should all be contributing a little bit at every point. But in clonal hematopoiesis, you basically get clonal expansions of individual hematopoietic stem cells in the bone marrow that then end up contributing disproportionately to the peripheral blood. And the way you can pick up this condition is that if you sequence peripheral blood samples, that you can see somatic mutations pop up at unusual allele frequencies in the sequencing data. So for our typical heterozygous germline variant, we would expect it to be in the sequencing reads at about 50% of the time. If we see mutations that pop up at smaller allele frequencies, then those would be indicative of a clone that's expanding in the blood. And what has been shown now across many studies is that this condition becomes more and more frequent as people age. So depending on how you define clonal hematopoiesis, by the time you're 70 or older, there may be 10 to 50% of all people may have such clonal expansions in the blood. And perhaps unsurprisingly, it was early on shown that clonal hematopoiesis associates with an increased risk of cancer in the hematopoietic system. So you're more likely to develop acute myeloid leukemia or AML if you have this condition. But what was more surprising and has since really engendered a lot of interest in the community were other associations that were consistently described with this condition of clonal hematopoiesis. So in particular, cardiovascular disease. There's many studies on population scale databases that have shown that various flavors of cardiovascular disease, including coronary heart disease, early onset myocardial infarction, atrial arrhythmia, and others, are associated with this clonal expansion in the blood. And so at least initially, people were very confused by this. Wow, why is that? And, this table here just shows what the hazard ratios are. So if you have a mutation that's detectable, as a somatic variant in your peripheral blood, your likelihood of being diagnosed with coronary heart disease is around twofold. And here in this case, for myocardial infarction, maybe even higher than that. And so people were initially very confused. Well, what does a clonal expansion in the blood have to do with cardiovascular disease? It's not obvious, and it immediately started investigating, as they should, as all scientists should, and really the primary point of investigation was the causal arrow from the mutant cells to cardiovascular disease. So do the mutant cells that people have in their bloods cause cardiovascular disease? And I'll just say that this is really like the classical causal hypothesis that would also underlie a lot of germline genetics. Mutation causes phenotype causes disease. And so here in this case, there were a number of papers, I'm just showing one example, that looked at what happens if you introduce mutations in genes that are often found in clonal hematopoiesis into mice. And indeed, these studies were able to show that, for example, TET2 mutations, really seem to confer a acceleration of atherosclerosis in a mouse model of cardiovascular disease. So if you give TET2 mutant cells into the blood of mice, you're going to end up seeing an increased incidence and increased severity of cardiovascular disease in a mouse model that's prone to develop this disease in the first place. All right, so all good. Somatic mutations may affect the tissue's function and cause diseases. That's really the classical causal arrow, and we can all be on board with that. But we also have to ask, because this is somatic genetics and not germline genetics, the inverse question, which is, well, but could it also be that the association is actually due to the disease causing the mutation's emergence? Is that another possibility that we have to look into? And we really became interested in this really early on, in around 2019 or so, reading the papers that came out at the time. And one interesting observation was that the risk of cardiovascular disease that appears to be conferred by mutations in different genes in the blood was really very comparable, for development of coronary heart disease. So here you see mutations in DNMT3A in your blood confer approximately 1.7-fold increased risk for coronary heart disease. Mutations in TET2, the hazard ratio is around 2. ASXL1, a different gene, around 2. A bucket of about 70 other genes that are all very different from each other. Again, same hazard ratio. And so this kind of made us think a little bit, because typically we think of one particular mutation having one particular phenotype. And so it's just surprising to see so many different mutations having similar phenotypes. They all seem to, confer an increased risk of cardiovascular disease. And so the other piece of information you need to know is that hematopoietic activity is actually, chronically increased in atherosclerosis. And I won't go into too much detail here because it's all, very kind of biological and kind of niche data that maybe not everybody will be interested in. But in mouse models of atherosclerosis and also in humans with atherosclerosis, it had been demonstrated over and over again that there's chronically increased hematopoiesis. That is because atherosclerosis is a chronic inflammatory condition. There's a low-grade inflammatory signal that these people experience for decades sometimes, and it causes the hematopoietic stem cells to turn over a little bit faster. The same goes for myocardial infarction. So this is now again shown very nicely in a mouse model here that, for example, 48 hours after myocardial infarction, hematopoietic stem cells start cycling because they have sensed that there's a danger in the body and they need to start producing cells to try and do something about it. And so here we really come to the core argument or core slide that, if you're going to remember nothing but one slide from this part of the talk, I hope it will be this one. Where I just want to remind you, and for many of you who work in population genetics, and this will come as absolutely no surprise. In fact, it's a bit of a trivial concept, but I'll still explain it in detail. And this concept is that if you have a variant that confers a selective advantage in a population, the rate at which this variant will expand in said population is dependent really on two things. It's the selective advantage, so the precise boost that the variant gives you, and second, the generation time, so how quickly new individuals are created in this population. So in hematopoietic stem cells in particular, imagine that you have two people in their hematopoietic stem cells. There is a small clone that's characterized by a mutation in a particular gene that confers a selective advantage. This clone exists at a variant allele frequency of 1%. Now imagine this stem cell pool over the course of 1 year divides 4 times. If the mutation gives you an advantage of about 15%, the VAF at the end of that year will be 1.7%. However, if you have chronically increased activity in the system, and in the same chronological year, stem cells divide 9 times, the VAF will be 3.5% because the mutation has been given the ability to manifest its advantage many more times, so to speak. So the rate at which mutations expand in our tissues absolutely depends on the selective coefficient that's associated with that mutation, but it's also really heavily dependent on the baseline proliferation rate that exists in the tissue. And so the question now, of course, is, well, if people with atherosclerosis experience chronically accelerated turnover of their hematopoietic stem cells, does that basically just lead to an accelerated expansion of the mutations that we all have anyway? In this case, the causality would be you're sick, hence your mutations that you have become visible more quickly. So it's a really different type of causal hypothesis than the mutations make you sick because the cells that are in your peripheral blood and that are mutated are damaging your cardiovascular system. So I'll just briefly.
Kamila, I
Go
have
ahead.
just a quick question. So is the underlying thinking here that these mutations do have some selective advantage, but without increased division would not rise to these frequencies? Or do you think that these are neutral mutations and just by drift reach higher frequencies when there's more division?
Yeah, it's a great question. And in fact, so in our paper that if people are interested can look up, we did treat both scenarios. So we looked at selection and neutral evolution. In both cases, increased cell division, of course, accelerates the rate at which any one given variant reaches a detectable level. So both for a neutral and for a positively selected variant. We know in the blood that the majority of the mutations really that people are interested in do have a selective advantage. So that's been corroborated in many different ways. But absolutely, through drift, you would get the same effect. It's just much slower. And the hematopoietic stem cell pool is still like, it's not that big, but it's also not nothing, so 100,000. But you do get, increased rate of detection for embryonic variants that are reaching detectable levels through drift if you have accelerated proliferation too. But it's a really good point. In fact, we spent years thinking about the neutral evolution aspect also. And that is in the paper, but I think that for most of the variants that people are interested in, there's pretty good evidence that they are positively selected. Yeah, great point. So I'll go a little bit more quickly now because I really wanted to focus just on the conceptual explanation behind all this. And now I'm going to show you some data that we collected to try and convince ourselves that our hypothesis, was plausible. But I'll go through the data a little bit more quickly. So the first thing that we had to figure out was, does atherosclerosis actually lead to an increase of proliferation at the highest level of the hematopoietic hierarchy? So in the hematopoietic stem cells, that's really crucial because in order to get clonal expansions that really grow over like an entire lifetime of a patient, they have to exist at the highest level of the stem cell hierarchy. If they exist at lower levels, those cells are short-lived and they get washed out of the system. So increased proliferation at lower levels wouldn't lead to these types of effects. So we had to really check, does atherosclerosis increase long-term hematopoietic stem cell proliferation and not just progenitor and the peripheral blood progenitor proliferation? And so we did this experiment in a mouse model, ApoE mice that are prone to developing atherosclerosis on an atherogenic diet. So they were given either regular diet or atherogenic diet. And then, without going into the details here, just want to show you this plot that quantifies the proliferation rate of hematopoietic stem cells. So if they divide, they incorporate this molecule BRDU. And so what you can see here is that if the mice are fed an atherogenic diet, indeed, their hematopoietic stem cells are proliferating faster, and that's a significant effect. So that's good. So we were able to show this in mice. Of course, mice are not the end of the story. We do have to convince ourselves the same thing is happening in humans too. And so this was, a big effort to try and get the right samples. And we were able to get samples from the sternum of control patients and patients who had active atherosclerosis. And we were able to look at the proliferation of HSC through a staining for a marker called Ki67, and saw that it was significantly higher in the people with atherosclerosis. So, and we could also get a quantitation of that, so about 2.4-fold higher proliferation in people with atherosclerosis. And so, as you know, you will know better than me, this kind of parameter is crucial for a mathematician because we have to know what is the precise increase in the proliferation of people with atherosclerosis in order to do a mathematical model. So what we now want to know, this observed increase of 2.4-fold, is that actually enough to result in a measurable increase of detectable clonal hematopoiesis at a given age? And so we set up a very simple mathematical model of the hematopoietic system that's based on the Moran process. So that's a constant population of about 100 hematopoietic stem cells that turns over. They can divide and create two new stem cells that live in the pool. They can divide and create two differentiated cells that drop out of the pool, or they can undergo asymmetric division, which is not explicitly modeled, as it doesn't, at least in the case where we're looking at positive selection, it doesn't change variant allele frequencies. And so, going further here. So essentially, what you can then model is that if you have a clone, at a given age, HT, and it has a particular size, what happens over X years if the baseline proliferation rate, which is B here, is a particular value to a mutation with the selective advantage S. And so relatively simple to do. One thing that we have to know, of course, is at what rate do clones in the blood typically grow and how big are they? Typically, at a kind of like a reference age, let's say 40 or 50 years old. So for this, we analyzed a bunch of different data sets and were able to figure out that, typically these clones in people grow at a rate about 4% per year. And so this 4% is either the product of the selective advantage and the baseline proliferation rate. But so we from this alone, you can't figure out what the individual values are, but you can figure out at what rate they grow per year. And what you can then do, since we know that there's approximately twofold or 2.5-fold increase in proliferation in people with atherosclerosis, you can basically now model, okay, how much faster do people with this increased atherosclerosis associated proliferation develop clonal hematopoiesis? And here you see that essentially the faster your stem cells proliferate, meaning the sicker you are, the faster they proliferate, the earlier clonal hematopoiesis will be detectable. So, for example, here in a person with healthy baseline proliferation, according to the literature, about one stem cell division per year, one symmetric self-renewal division per year, typical such person would get clonal hematopoiesis just when they're around 80 years old. But if your proliferation rate, your baseline proliferation rate is increased threefold, you will have clonal hematopoiesis by age of 55 already. So it's a real difference. We also, of course, looked at the relevant variance in proliferation rates, because not everybody gets a perfectly threefold increase in their baseline proliferation. So we modeled the variance in proliferation rates that we observed in our data set, and we really looked at what kind of effects could this plausibly result in. And what you can see here is that if you have atherosclerosis associated proliferation rates, then your likelihood of being diagnosed with clonal hematopoiesis by age 70, is about 3.5-fold higher, or your risk ratio is about 3.5-fold higher than the control. So this is pretty comparable.
We have a question from the chat that I just saw. And the question is 100 hematopoietic stem cells a realistic number for humans?
100,000.
Because I think the first... Okay, yes. I think the first time you said 100,000 and the second time you said 100. So it's definitely in the simulation also 100,000.
Oh, yeah, absolutely. Sorry. So I sometimes just eat my words. I may have also used 100 as just like a illustrative example, but it is 100,000. And also in the simulation, it would be 100,000. I will say, though, the simulation doesn't, so the simple simulation that you see here actually doesn't explicitly model the number of cells. It begins with a clone that has a particular frequency. So for example, it has a frequency of, I can't really see here what the axis is, a certain frequency below 1% at the age of 40. And then it grows exponentially at a certain rate. And so this exponential growth is now just accelerated. But because we're looking at positive selection, we're not modeling neutral evolution and drift. This is actually kind of independent of the stem cell pool size, if that makes sense.
Okay, yeah. Thanks.
Okay, thank you so much. Yeah, 100,000 stem cells. All right, great. So I'll go a little bit quicker now because I still have one other interesting, surprising causation story for you, but I'll just very briefly say, you do sensitivity analysis, so you look at different division times for the baseline proliferation rate, you look at different driver effects. Actually turns out that the increased risk ratio for clonal hematopoiesis at age 70 is incredibly robust. Like you're always at around 3, which is now very consistent with like the actual data we see where, all these different genes appear to confer about the same risk. And that's really, consistent with, just looking at different values on the y-axis here. So you could have a gene that has, selective coefficient of 8, and you will have 8%, sorry. And you will have a risk ratio of about 2.5, or you have one that's significantly weaker, and you will still end up in the same area. So this was always very interesting to me that I think this actually explains pretty nicely what this data set here shows. All right, so very briefly, and I'll skip a bunch of slides in a moment, but this is the crucial experiment I now want to show you. Unfortunately, when it comes to like pushing kind of a counter narrative, people are usually don't it doesn't suffice to just show theoretically that it kind of has to work this way. You have to also demonstrate it. So we had to go and prove in mice that this was really true. So what we did is we admixed TET2 mutant bone marrow, so that corresponds to the clone with a selective advantage, into mice that were atherosclerosis prone. And then, as in the previous experiment I told you about, they were put on either a chow diet where there was very slow atherosclerosis development, or on an atherogenic diet where they develop pretty rampant atherosclerosis pretty quickly. And of course, the prediction of the mathematical model is, and I won't go through this in detail, I'll just maybe show one of these curves, for a given fitness advantage of the TET2 mutant cells, you should be seeing that the TET2 mutant fraction in the hematopoietic stem cell compartment and also in the peripheral blood rises more quickly, because there is a quicker baseline proliferation. So the advantage of the mutation is more, has more opportunity to manifest itself. So that's the theoretical prediction, and this is the real data. Indeed, if we do this, we check the frequency of the mutant cells in the peripheral blood now in monocytes and neutrophils in the two blood types that are most short-lived and of interest in this setting. You can see that in blue, indeed, that's the mutant fraction. You can see it, in blue, that's the atherosclerosis mice. So the blue curve shows the mutant fraction in the mouse cohort that got the atherogenic diet that has a higher baseline proliferation rate, and the mutation, in fact, expands faster than in the mice that are on chow diet, and that's very consistent between monocytes and neutrophils. So very nice confirmation, and in the end, when we sacrificed the mice and we looked at the normalized TET2 fraction as a normalized to baseline in the atherosclerosis cohort, we could see, yes, indeed, also in the LSK, which is a progenitor compartment, and in the hematopoietic stem cell compartment, these mutant cells were expanded more. I'll just very briefly say that this has been shown to happen in patients too. So this is a very nice study that came out a few years ago that shows that the clone growth rate, if you measure the clone growth rate longitudinally in people, that it negatively correlates with markers of cardiovascular risk. I won't go into too much detail, but if you're interested in looking it up, this is the paper, and really nice to see this in humans as well. We did it in other scenarios also. So anything that increases your proliferation rate really increases the rate at which mutations in your blood expand. So same happens if you deprive mice of sleep. Turns out that's really bad for them. And so their hematopoietic stem cells also proliferate faster. You can see that the mutant cells also expand faster in that setting. All right, I'm going to skip over this and just leave you with the take-home message here, that I think that there's an interesting proposition for causality that's being made here, namely that it's not necessarily the mutant cells that give you cardiovascular disease, but it's cardiovascular disease that also increases the fraction of the mutant cells in your bone marrow. And again, I want to be very clear that I'm not saying the mutant cells don't then do something on their own too. It's just that both things may exist at the same time, and we can't take a particular causality for granted the way we do in germline genetics. And really the take-home message really is that anything that increases the proliferation of hematopoietic stem cells, or really any stem cells in any tissue, really for what it's worth, is expected to lead to an accelerated rate of clonal expansion. All right, great. So if there are any questions on this part, we could very much, do them now. I'll switch gears a little bit and, use another 15 minutes or so to tell you about a different story. But if there are any questions on this part from you, Ivana, or anybody else, then.
No, I'm good. And because we asked some questions already during the talk, you can take 20 minutes if needed.
Okay, perfect. All right. So different topic now, but again, it's going to be in the vein of how do we really distinguish between causation and correlation in somatic genetics. And so this talk is going to be about a question that seems really easy to answer on the surface of it, but I think that you'll see that maybe it's not so easy to answer. And this is all the work of a really fantastic postdoc in my lab, David Cheek, who's a trained mathematician and probabilist who a few years ago decided to join the dark forces in a more biology and genetic centric lab, and it's been really wonderful to have him. And he's really the driving force behind all of this. And this project is his brainchild, and I'll be very happy to tell you what this brain came up with. So, which mutations cause cancer? Seems like a really trivial question. Haven't we sequenced so many cancer genomes? Don't we know which mutations cause cancer? Indeed, we have sequenced many cancer genomes, and we have come up with a list of cancer driver mutations that typically, are derived from pretty sophisticated but conceptually simple measures of positive selection. So, for example, we take a bunch of cancer genomes. We look for a given gene like p53 at the number of non-synonymous mutations that we find in that gene. We compare it to the number of synonymous mutations that presumably reflect the baseline kind of background mutation rate at that particular locus. And then we look at how these two measures compare to each other. And those genes that have great preponderance of non-synonymous mutations, we really think of as being under positive selection in cancer formation. And that's, of course, worked very well. I mean, many of these are, really famous cancer driver genes that a lot of therapeutics are being developed around, et cetera. However, there's a little bit of a but, and that has really only emerged in the last 10 years or so, as we have figured out how to do sequencing of normal tissues in a better way. So for the longest time, people have thought of the genomes of cells that sit in our normal tissues as these like beautiful, pristine, wild type cells, that have these wonderfully normal genomes, and the cancers have such a barren genomes. So, but as it turns out, if you just figure out how to sequence very small populations of cells in our normal tissues and how to do this in a way that's properly error corrected, you will find that actually there's so many mutations in all of our cells, and many of them are under positive selection. So mutations arise during embryonic development, during childhood, then during the turnover of our normal tissues. Some of these mutations will have a selective advantage, and they will expand locally to form small microscopic clones. And as I mentioned earlier, it's really on the backdrop of that clonal evolution in our normal tissues that carcinogenesis is happening. And so we have to ask, well, if we find a particular mutation in the cancer, is it really there because it causes cancer? Or is it maybe there because it was selected during this process of normal tissue evolution over time? So to just repeat the question, that's going to be the question that will drive us for the next 10 minutes or so. Do signals of positive selection in cancer genomes reflect cancer causation as they have traditionally been assumed? Or are they, at least in some cases, merely an inheritance from normal tissue evolution? So we can think of it as a Venn diagram. There are some mutations that are under positive selection in normal tissues. There are some mutations that cause cancer. There's going to be some overlap between the two. But how large this overlap is, and in which of these three buckets any one given mutation falls, we don't know yet. So how do we even approach this question? How can we try to really begin to think about separating selection in normal tissue from cancer causation? So what David came up with, because he is a probabilist, is a quantitative definition of carcinogenicity that I think is very different from typical cancer driver mutation definition. And it is that a cell with a particular mutation Z causes cancer initiation. So it causes the emergence of a malignant lineage that will eventually at some point be diagnosed as a cancer at a rate R1. And then a cell without that same mutation Z causes cancer formation at rate R0. And he really calls the mutation Z's carcinogenic effect just the ratio between these two rates. So R1 divided by R0, also written as W, is the mutation's carcinogenic effect. And the nice thing about this carcinogenic effect is that it can be estimated from data. So we really actually only need two pieces of information. One is harder to get than the other, but they're both increasingly available. So we need Y, which is the frequency of that mutation among cancers of that type. So let's walk through an example. For example, esophageal squamous cell carcinoma, we have sequenced many esophageal squamous cell carcinomas. You can look it up in TCGA or in your preferred cancer genome database. And we can get the frequency, so the number of tumors that contain a mutation in that gene. And then we need X, and that's the frequency of the mutation among normal cells in the same tissue of origin. So a regular esophageal epithelial cell, at what frequency is it mutated for the same gene? And for a long time, this data wasn't available, but increasingly it now is. So for example, this paper that came out in Nature 2019 took very small samples from the normal esophageal epithelium of people who were undergoing cancer surgery, and they were deeply sequenced, and we can get the frequencies for all these like famous cancer driver mutations in that normal esophageal epithelium. And so what we can then do is use these frequencies to estimate the carcinogenic effect. So the carcinogenic effect is estimated as the odds ratio of the frequencies in the cancer versus the normal tissue. And we can plot them in this type of very nice plot that David pioneered, where really it's a very simple plot if you think about it, but it gives you a very unique view. On the x-axis, we're looking at the mutation frequency in normal tissue, and on the y-axis in the mutation frequency among cancers. And mutations that are much more common in cancers but very infrequent in the normal tissue we think of as carcinogenic. However, mutations that are very frequent in normal tissue but less so in the corresponding cancer we think actually of as cancer inhibiting. And then everything that's along this diagonal here is neutral with respect to cancer formation. And so if we now do this, we actually take the real data from esophageal squamous cell carcinoma, TCGA-derived frequencies, and from sequencing of normal esophageal epithelium, and we put the genes onto this plot. Now we'll just say all the genes that you're seeing here are what used to be canonical esophageal squamous cell carcinoma drivers, so genes that were famous for being under suspicion of causing esophageal squamous cell carcinoma. What you can see is some pretty interesting things. So for example, Notch1, which is among the genes that are most frequently mutated in this particular cancer type, turns out it's actually cancer inhibiting because it is even more frequent in the normal esophageal epithelium than it is in the corresponding cancer. There are, of course, also counterexamples. No surprise here, for example, P53, much more common in the cancer than in the normal esophageal epithelium. But there are also quite a few interesting genes that are along this diagonal here. So carcinogenic effect 1, meaning that they have no measurable effect in this data set on the probability that a cell will found a malignant lineage. Some of these are famous mutations like PIK3CA, FAT1. And so this particular way of looking at the world really, I think, makes us think in a new way, or makes us interpret the genes that we find to be mutated in cancer in a new way. So we can now really do this for every tissue that we have sufficient data for. So to return to the hematopoietic system view, it's a similar view where we're looking at classical cancer drivers of AML, acute myeloid leukemia. And we can see that the most common genes that are mutated in AML, including DNMT3A, TET2, ASXL1, actually have very moderate carcinogenic effects, meaning that they don't really increase the likelihood of that lineage forming a cancer that highly in comparison with some of the other genes like FLT3 or IDH2 mutations that have carcinogenic effects that are there in the thousands.
And sorry,
All
do
right.
you see?
Yeah.
In the blood, do you see something in the gray area, like Notch1 in esophagus, or is everything kind of selecting for cancer?
Well, thank you for this question, because it brings me to the next topic very naturally. So it's really interesting, right? So here there's a lot more green than there is here. And so this could be that maybe the blood just works differently. But one really important difference between these two slides is also that in the case of the esophagus, we actually had esophageal tissue that came from the same patients that also had esophageal cancer. while in this slide, the control is normal blood from people who don't have leukemia. And so this brings me, to this next slide. Selective pressures in normal tissue can be influenced by a lot of things, including germline variants, lifestyle factors, like we just talked about, right, in the clonal hematopoiesis section, in the previous section. How you smoke, what you eat, how sick you are, it's really going to determine the rate of evolution and maybe some of the variants that are selected for in your tissues. So it could very well be that the overall somatic landscape in matched normal tissue samples from cancer patients looks very different from the landscape in people who don't have cancer. And it's actually a really interesting question, which one of the comparisons is the more useful or more appropriate one. It's something that we're talking about a lot. Like, I think that ideally you would want the data to come from cancer patients, but from lineages that have not turned malignant. But of course, that's even more difficult, right? So we have a lot of normal blood sequencing data that we can use to kind of estimate the frequencies of these mutations in normal blood. But getting the same type of data from lineages in people with AML that are not AML, much harder to do. So this is, really one of the big hurdles to getting good carcinogenic effects for many types of cancer, is that, sort of suitable normal tissue data is just really hard to obtain. So David had a really, really cool idea that, I immediately was extremely enthusiastic about when he came up with it. Namely, he asked himself, can we identify carcinogenic mutations from cancer genomes alone without having the matched normal tissue data? Now, so that's a really severe case of bootstrapping, because you really need the normal tissue data in order to make sense of these carcinogenic effects. But I'll just briefly, and that's really where I'll be focusing, and then I'll point you to David's bioRxiv paper so that you can, look at your favorite cancer types and genes and what their carcinogenic effects are. But I'll just explain the concept of the idea that he came up with. And that's namely, let me just take a step back, and we'll go on a small tangent where you consider with me what happens with mutations that are positively selected in normal tissues yet don't cause cancer. Okay, so let's think about it. What happens if you have a mutation that arises early on in your tissue? If it has a large selective effect, meaning that it is positively selected, then it will be growing over time. So in older people, you will find more mutations that are expanded in the tissue because evolution has had time to act on the mutant cells and expand them to appreciable frequencies. And so if you now look at the cancers that arise from these background tissues of people with different ages, you will see that in older people, you will be more likely to have an inheritance of these mutations that were selected during normal tissue evolution to the corresponding cancer, and less so in younger people. And if you now consider a mutation that has a carcinogenic effect, so that's not just positively selected, but neutral with respect to cancer formation, but has a real carcinogenic effect in the sense that it highly increases a cell's likelihood of becoming a diagnosable cancer, you will see that the expected fraction of cancers with that mutation has a really different shape from the distribution or from the curve that shows the mutations that simply have a selective normal advantage in normal tissue. And you will see that these two curves overlap for older people. So I'll just say it in simpler words. When you have a cancer in an older person, you don't really know, is the mutation in that cancer because it was selected during normal tissue evolution and then inherited, or is it there because it's carcinogenic? But in younger people, you can, because in younger people, it's very unlikely that a mutation will be there due to normal tissue evolution. Young people haven't had time to expand mutations in their tissues due to regular, positive selection during normal homeostasis. And so if there's a high frequency of a particular mutation in cancers that come from young people, that's much more consistent with a real causal effect, so a real carcinogenic effect of that mutation. So, to just show you very briefly what this looks like in AML. In AML, for example, we know that different types of mutations have very different age distributions. So KIT, for example, is often mutated in young people. TET2 is often mutated in older people. Our hypothesis here now is that selection during normal tissue evolution and carcinogenesis interact to create these very different age distributions. So TET2 would be more frequent in older people because TET2 is selected during normal tissue evolution, hence more likely to be associated with a cancer that arises in older people. The alternative hypothesis, of course, that this is just age-varying biology. So a KIT mutation, for example, will give you AML if it happens in a young hematopoietic stem cell, but in an older hematopoietic stem cells, due to the different biology of these cells, it won't give you AML. We're not disputing that. That could still be the case, but let's see to what degree the first explanation actually holds. And so in order to do this, I'll show you the data in a slightly different way. So if we just look at the frequency of the most common AML driver mutations in young people who get AML versus their mutation frequency in AML that comes from older patients, you will see that there's basically no relationship. So there's basically no correlation, very different genetic profiles between young and old onset AML. But if we now plot this data differently, namely on the y-axis, we don't plot the mutation frequency in older onset AML, but we plot the estimated carcinogenic effect, which is what I showed you a few slides earlier, where Ivana asked, oh, there's so much green. Those carcinogenic effects where we're normalizing the mutation frequency in the cancer by the corresponding normal blood frequency. And we plot this against the estimated relative carcinogenic effect for young onset AML, which is essentially just the gene length, because we assume that in young people, evolution hasn't had time to expand mutations, so the mutation frequency is just proportional to the size of the gene. If we plot these against each other, you can now see that it does all of a sudden line up. So in simple words, I'll just say it again in simple words, that if you normalize these mutation frequencies in older AML patients for the degree to which the gene is selected in normal tissue, you actually end up all of a sudden finding agreement between how carcinogenic mutations are in young and in old people. So the difference actually isn't so big between young and older onset AML, if that makes sense. I saw that Ivana unmuted herself, so maybe it doesn't make sense yet, but please ask questions.
No, it seems that we are having some technical difficulties with, so you were frozen on my side for a second, but we can talk more about this during questions. If
okay.
It's unclear.
Sounds great. Yeah, let's finish up now so that we still have a little bit of time. So I'll just say very briefly, these carcinogenic effects that Dave is estimating for AML, they're very consistent with more classical ways of estimating the hazard for AML that's associated with certain mutations. And I encourage you to look it up in the paper. I'll show you the reference in just a moment. I'll just say that the carcinogenic effects, generally we can correlate with mean age. So in the tissues where we can actually do carcinogenic effect estimation, we can see that indeed the mean patient age is highly correlated with the carcinogenic effect that we get through comparison to normal tissues. And then finally, for those tissues where we just have no information whatsoever, because there's no normal tissue sequencing data available, we can still do this trick and look at mutations that are preferentially mutated in younger patients and are at the same time under positive selection in cancer. And for those, we have strong suspicion that those are truly carcinogenic. All right, so this is the end, the summary that hopefully you can remember from this part of the talk. Positive selection in normal tissues can really distort our view of cancer causation. And I think it's a really fascinating research area as more and more normal tissue sequencing data appears. I think that we'll all be studying this and wondering what it really tells us about the cancer genome that we thought we knew so well. And the genomes of young cancer patients can provide pretty clear views of cancer causation. And so unfortunately, age data is not always collected in these big databases. I hope that maybe this will change, and the age information that is more of a niche research area, I would say, maybe becomes more important in the light of these very principled considerations. So if you're interested in any of this, David's paper, it's called Age Distinguished Selection from Causation in Cancer Genomes. You can look it up. Yeah, I'll end here and just acknowledge all our collaborators. This is just kind of the standard lab slide, but in particular for the clonal hematopoiesis story, we worked very closely with the labs of Phil Swirsky, Matthias Narendorf, Martin Novak. It was a real team effort, and so those, of course, are all acknowledged as well. And thank you for your attention.
Thanks a lot, Kamila. This was really, really interesting. I have a bunch of questions, but let's see if there are any questions from the audience first. So please, if you have any questions, type them in the chat. And maybe while we're giving time for people to type in their questions, I think this is a very neat idea of trying to disentangle selection versus just age-related effects, or selection in normal tissues versus selection for cancer. Did you think about that maybe mutations that you see in younger patients could somehow have more to do with development, or would you actually see those both in older and younger patients? So basically, like playing the devil's advocate, what are the things that could mess up your logic?
Oh, many. I mean, biology is just made to mess up any logic, right? So I think the important part, and this actually came up as like a big point during review too, and I do want to be really clear about it, that this type of framework, it doesn't negate the possibility of like age-specific carcinogenic effects. So I think nobody wants to deny that there are some mutations, in particular, as you said, mutations in genes that are relevant in development, that often are really specific to childhood cancers. And childhood cancers also often arise in organs that are really different, and they absolutely do have a different biology. I would say that the ideal data for this kind of analysis to me is not, the types of cancers that arise in a newborn versus, an 80-year-old. I think what we can really take advantage of with just the right patient cohorts is younger adults, like somebody in their early 40s versus older adults, somebody who's maybe in their 80s, where we do think that the biology of the tissues, is pretty comparable. And with just, enough data, because of course, cancer in younger people is more rare. But that's actually, I'll say this is a kind of another PR point. We should be sequencing the genomes of young cancer patients. So not just because everybody's interested in young onset cancer. These genomes are particularly pristine in the sense that they contain less kind of inheritance from normal tissue evolution. They give us a clearer view of what actually causes cancer. And so younger patients, not necessarily children, but younger patients for any given cancer type contain this valuable information and should be sequenced. And if we have enough data, then even like a mutation that's enriched in people who are only 5 years younger or so, I think can already give clues. So I think that's probably the best way of thinking about it, if that makes sense.
Yeah, and we have a couple of questions from the chat. So the first one is, does clonal hematopoiesis always start from a hematopoietic stem cell, or you may have the start of a clone downstream in hematopoiesis?
Yeah, very good question. I think that's absolutely possible that clones start downstream. Typically, unless they turn into cancer, they wouldn't be so long-lived. So not that everything is understood about normal hematopoiesis, but we really think of the hematopoietic stem cell as being the population that's the only long-lived enough population to allow for clone expansion over decades and decades. All the clones that happen at lower hierarchy levels could temporarily exist and persist and maybe even be detectable but they wouldn't be inherited over a really long time frame, just because these cells kind of die away and they're always replenished from the top. So for really large clones, it's just most likely that they come from the hematopoietic stem cells, because these are the only ones that are long-lived enough to allow for clone expansion that becomes really big.
And another question is, what is the age cutoff of young and old patients? How is this threshold being determined?
Yeah, there is no good cutoff. We tried, so for the analysis that you see, we tried just a bunch of different ones, and we showed that it's always robust to that. So if you want to look in the paper, at some point you have to make some definitions. But in the plot that I showed, the average age of the young onset AML was 20, and the average age of the older onset AML was 60. But they're both distributions. But we did look at this pretty carefully to make sure that the results weren't contingent on one particular cutoff, which would be kind of not so reassuring. But they were very robust.
Well, thank you so much. It was really a pleasure having you, and thank you all for watching.
Thank you. Thank you so much for your time.
Great. Thank you so much for